Hey everyone, and welcome back to our MBBS with AI Biochemistry series. Today, we're tackling a topic that's not just important, but is one of the most high-yield exam favorite subjects in all of metabolism. We're talking about oxidative phosphorylation, which you probably know better as the electron transport chain. We start with glucose, which undergoes glycolysis to produce Now, pyruvate is at a major metabolic crossroads. It could go back to form glucose through gluconeogenesis, but its most common path is to be converted into acetyl-CoA. This acetyl-CoA then enters the powerhouse of the cell's metabolic pathways, the TCA cycle, also known as the Krebs cycle. The real magic of the TCA cycle isn't just about breaking down molecules. Its true purpose is to generate NAD and FAD. Think of these molecules as fully charged batteries, packed with high-energy electrons. The entire purpose of today's video is to explain what happens when we plug these batteries in. So, where do these electron carriers go? NAD and FADB travel to the inner mitochondrial membrane, the site of oxidative phosphorylation. To visualize this, imagine the inner mitochondrial membrane as a long, folded barrier. On one side, you have the mitochondrial matrix, which is the innermost compartment of the mitochondrion. On the other side, you have the intermembrane space, the area between the inner and outer mitochondrial membranes. Embedded within this inner membrane are 5 major protein complexes, which are the key players in our story. Let's follow the journey of the electrons. It all starts with NADH, which arrives at complex I and hands off its high-energy electrons. Meanwhile, FADH takes a slightly different route. It delivers its electrons to complex 2. Now, here's a crucial point. Both complex 1 and complex 2 then pass their newly acquired electrons to a small, mobile carrier molecule floating within the membrane called coenzyme Q, or CoQ. Think of coenzyme Q as a shuttle bus, picking up electrons from both starting points and carrying them to the next stop. From coenzyme Q, the electrons are transferred to complex 3. After complex 3, they hop onto another mobile carrier, a protein called cytochrome C. So, the path is from NADH2 complex I4, FADH2 complex 2, then both to coenzyme Q, then to complex 3, to cytochrome C, and finally to complex 4. The energy released from the electron's movement is used to actively pump protons, which are just hydrogen ions from the mitochondrial matrix, across the inner membrane and into the intermembrane space. Imagine using the flow of a river to power a pump that moves water uphill into a reservoir. Each time electrons pass through these specific complexes, more and more protons get pumped into that intermembrane space, creating a powerful electrochemical gradient. Proton gradient is like a dam holding back a massive amount of potential energy. Now, you might have noticed I skipped over complex 2 when talking about proton pumping. This is a super important detail and a common exam question. While complex 2 is part of the chain because it accepts electrons from SADB, it does not pump any protons. This is the fundamental reason why SADB ultimately generates less ATP than NADH, because it contributes less to building that proton gradient. So, what happens at the very end of this electron journey? At complex 4, the electrons have lost most of their energy. They need a final destination, a place to rest. This is where oxygen comes in. Oxygen is the final electron acceptor. It graciously takes these low-energy electrons from complex 4, and at the same time, it combines with some of the protons from the matrix to form water. This fact that oxygen is the final electron acceptor and that water is formed is extremely high-yield information. All those protons are crowded into the intermembrane space, desperate to flow back down into the matrix where their concentration is lower. ATP synthase is a remarkable molecular machine, almost like a turbine in a hydroelectric dam. This elegant process, where the movement of ions across a membrane is coupled to ATP synthesis, is called chemiosmosis. It's oxidative because it involves the transfer of electrons, and it's phosphorylation because we're adding a phosphate to ADP. For your exams and clinical understanding, it's absolutely vital to know the substances that can inhibit this finely tuned system. Let's run through them. Rotenone, a common pesticide, specifically inhibits complex I antimycin, A, an antibiotic. Blocks complex 3. Oligomycin, another antibiotic, works differently. It directly blocks the proton channel of ATP synthase, so even if you have a gradient, you can't make ATP. Finally, there are uncouplers like the classic example 24-dinitrophenol, or 2, for DNP. Uncouplers essentially poke holes in the inner mitochondrial membrane, allowing protons to leak back into the matrix without going through ATP, and the energy from the gradient is still released. But instead of making ATP, it's dissipated as heat. So, let's put it all together and look at the final energy tally. Why is oxidative phosphorylation so dominant? Well, glycolysis, which happens in the cytoplasm, gives us a net gain of only 2 ATP. The TCA cycle, happening in the mitochondrial matrix, directly produces only about 2 ATP in the form of GTP. The real payoff comes from the NADH and FADH that were generated. Each NADH molecule that enters the electron transport chain yields approximately 2.5 ATP, and each FADB yields about 1.5 ATP. When you add up all the NAD and FADP produced from one molecule of glucose through glycolysis, pyruvate oxidation, and the TCA cycle, and then run them through oxidative phosphorylation, you get a grand total of somewhere between 30 to 32 ATP molecules per single molecule of glucose. This is why oxidative phosphorylation is rightfully called the powerhouse of cellular metabolism. It's the primary reason our cells can generate enough energy to power everything from muscle contraction to nerve impulses to DNA replication. And that is precisely why mastering this topic is absolutely non-negotiable for every single MBBS student. Thanks for sticking with me through this deep dive into the electron transport chain. I hope this explanation helps you visualize the process and lock in those high-yield facts for your exams. If you found this video helpful, be sure to hit the like button, subscribe to MBBS with AI, and turn on notifications so you don't miss our next lesson. See you in the next one.